Good afternoon, Dr. Arber. It's an honor and a pleasure to have you here. I have to mention a few words before getting started, which is that Dr. Werner Arber, who is the Nobel laureate in the 1978 Nobel laureate for medicine, is here invited by the Rey Jaime I Prize Foundation. And my name is Carlos Simón. I was awarded by the Rey Jaime I Prize in 2011 for medical research. So this is the introduction. And what I would like to do is, of course, to welcome you here. And as mentioned to you, I have a really, we are all very grateful for what you have done for the field. In fact, everybody in our laboratories, HINDE3, and all the restriction enzymes that you invented, they are the main source of, the main tool that we use in the lab. What I would like to know, I have now the opportunity to ask you directly, there are things that they are happening now, that they are being relating backwards to your work so long time ago. And as you may imagine, I want to ask your opinion about CRISPR, about all of these systems that nowadays is being used for gene editing with a very high promise. And if we go back, the scissors for that, they were invented back in your work. So it was you probably the real pioneer backwards. So first, what I would like to ask you is, what do you think? Do you foresee the potential of CRISPR for gene editing and going, later on we talk about the human situation, but what do you think about the technology?
Yes, I have given some attention to that development of knowledge on CRISPR. And I must say I was only a little bit surprised because I have seen in my past work always that nature is astonishingly inventive. They have various very different mechanisms to reach similar goals. And we have studied for the last few decades the generation of spontaneous mutants, which are, of course, the driving force of biological evolution. Without occasional mutation in one organism of a large population, there wouldn't be any evolution, it would always be the same. And our observation is that despite the fact that they use a number of enzymes, either to generate actively mutants or to keep mutation rates very low, because, of course, that shouldn't happen each generation, because otherwise the stability of the species would be endangered, because not all of novel mutations, by far not all novel mutations, give a selective advantage to those having suffered a mutation. So this insight, these are insights into the laws of nature to drive biological evolution of all living beings. We had done our research usually with single cellular bacteria and bacterial viruses, but it then became obvious that in principle the big concept of spontaneous mutagenesis and natural selection is true for all living beings on our planet.
So as you may read, very recently they have been trying to use these tools, let's say CRISPR talents, just to correct these mutations in mitochondrial DNA. They start with the mitochondrial DNA in order to see the easiest way to do it. But then the plan is to go to nuclear DNA for that, to actually correct, to edit this. What do you think that, what are the risks, or what do you think about the technology itself being applied to the humans?
Well, for me, there were other methods to make different kinds of mutations in the laboratory by purpose. Shortly after the restriction enzymes became available in the early 1970s, in part the same people who were working with these enzymes and their application also had designed methods for site-directed mutagenesis. This was occasionally applied, but the new methodology, which is based on the CRISPR system, may be more accurate, more efficient. So it is a fact. Now, the question is, if one goes into the germline, then one should, for me, distinguish between microorganisms, between plants, different kinds of plants, and animals, and then finally human beings. So in the field of microorganisms, I hope very much that people will not render bacteria more pathogenic, which in principle could be tried out. In the field, on the other edge, humans, if you change actively the human genome, you should accompany that by ethical considerations. I think that's important, and one doesn't let the scientists just do whatever they wanted to do.
Absolutely. And what do you think about the off-target effect of this technology? Because this is, for me, the main question mark. Because if you try to correct, to cut and paste a mutated gene, and then you have, off-target effect, and you are affecting another part of the genome, and then you produce a problem. Do you think that this is a reasonable doubt nowadays? What do you think about that?
Well, in principle, one can see whether before, or let's say the sequence of that segment of DNA which should suffer an alteration before and after carrying out the reaction. And then if after the reaction it's not what you expect, just that particular alteration which you intend to do, but some other things, then one shouldn't probably, one should discard that. This would be fair.
Yeah, you have to. Many people, they are worried about gene therapy because editing is about, we may want to really avoid disease, mutations that produce disease, which is, at least in my case, the main focus of this research. Yeah. But people are starting to be worried about a la carte children. And so, of course, there is this is one completely different scenario. Doctors, we think in human and in health, we think in diseases, not in the other. But as you say, there must be ethical considerations.
And we must find a consensus on rules to apply. Yes, that's important. Yeah.
That's because for the general public, they may fear sometimes the technology if they don't know good enough what is the mainstream of this technology to be used.
Yes.
Do you think that gene therapy is going to be possible in the future? In general terms, of course, to correct disease.
Well, I'm not sufficiently familiar with medical applications. I'm not an MD, and I worked all my life with bacterial genetics. But I would say, if you have already a person having a genetic defect, I would have a hard time to know in which cell an alteration should be made. That would help the patient, because you have so many cells in your body, and I'm not
No, but the whole idea will be to work on the embryos itself.
Yeah. Okay,
Or the moment that your embryo is, that is genetically abnormal,
Yeah.
and this will be the moment. Those technologies, they will act, CRISPR and TALENs, at the moment of pre-implantation, before transferring the embryo back to the mother. So by that, we expect to recover. This was the work with the mitochondrial DNA. So this will be the idea, to work on the embryo before implantation, trying to correct this. That's why that's the main concern about off-target therapies.
Well, okay, again, I'm not quite sure whether I can answer that correctly, but of course, a baby has two parents, and it is possible after in vitro fertilization to look at the chromosomes of embryo, of very early embryo, and the practice, if I'm well informed, usually a few embryos are made, and I think it's fair to select those which have not this particular mutation from the parent which may have inherited it. So that's a selection. You don't need to correct anything.
Yeah,
that
nowadays
would be the easy approach.
Nowadays, this is exactly what happens.
Yeah.
We work on selection of chromosomally or genetically normal embryos. But sometimes all the penetrance is so high that all the embryos are abnormal. So that's why it's an attempt in our field to look for gene therapy, gene editing, what is called now. In our case, for those parents that they cannot have their own genetic offspring because they know that a mutation is going to be there. So this is the case, and because of that, we foresee that this could be something interesting to prepare for the next, in those cases that they are impossible to have their own family. That's
Yes, I think this kind of question requires a serious discussion of the involved scientists with ethical specialists on the first hand, and secondly with the general public. I think not a small group of scientists and with some ethical people should make a final decision, but one has to have the consensus of the wider audience.
Yeah, absolutely. And I would like to ask you about all of these mechanisms and maybe and also very big important discoveries. For instance, penicillin. That has been based on what a microorganism is actually doing. So many things has been done sometimes by serendipity, and in this case of CRISPR is the mechanism, and you study this, is the mechanism by which a bacteria defend itself from the viruses.
Yes, and from from other species related to the one of the bacteria which is concerned. So we have really very seriously studied horizontal transfer. Originally, that was investigations, scientific research was done when the horizontal transfer of antibiotic resistances came up. And it is, when I was young, 50 years ago, I remember I had tried to, in my talk, to include that one should use less antibiotics for growing animals, cows, hens, which were fed at that moment every day antibiotics, and that's of course favoring enormously antibiotic resistances in these animals. And this is still nowadays being done, and we have the problem, particularly in hospitals, that horizontal transfer of antibiotic resistance between different microorganisms happens every day. So then the non-resistance pathogens all of a sudden have a subclone of pathogens. And this could be really much more avoided if not so many antibiotics would be fed to animals every day.
Yeah, but this is the general, this is the general belief nowadays, not to overload with antibiotics because the resistance is getting out of hand.
Yeah.
Absolutely. And what I mean is that so much to learn from those microorganisms that they, in order to survive, they are able to produce those mechanisms that we use to understand gene therapy, those mechanisms that we understand to penicillin, for instance, how to kill the fungus, for instance, this is, I think this is extraordinary, the amount of knowledge that we can gain from them.
Of course, when we started this research, we had no idea how it works. I had been, my PhD study was, among others, was studying a bacterial virus, bacteriophages, which had been grown in galactose plus bacteria, and some of these viruses had picked up the capacity to transfer horizontally, the capacity to ferment galactose, and in my electron microscopical and accompanied by genetic studies, I could show that these viruses had actually integrated into the genome of that virus a segment of the former host with galactose fermentation genes, and of course some genes of the virus had been deleted in order to still fitting the thing into a viral particle, and the missing genes were for building the capsule of the virus. So the capsule, the virus could still be grown in the presence of a non-mutated similar virus, and as a helper virus. And this idea, what I talk here, that was in the end of the 1950s, gave the idea that one knew already that the genomes, even of bacteria and of course of higher organisms much more, are tremendous long filaments of DNA. And people said, well, we cannot study individual genes on this long DNA. One has to sort them out. And that defective virus, having lost a few viral genes but put in that space bacterial genes, was an example. People felt that they would, what one should do is taking a virus, open it, including one or a few genes which you want to study, and then you can grow it up like a virus, and harvest that, and then make structural and functional studies. That was the idea.
This was the beginning, huh?
Right. But this only started to be easy when the first restriction enzymes became available. That was around 1970, so 10 years later. But the idea was there how one should do the genetic engineering, but it didn't work properly.
Yeah, but at the end it goes through. Yeah.
And there are other, of course, plasmids which also can be used as gene vectors to grow up. And that was very helpful in these early times.
Do you have any at that point, at that time, do you have any, bad opinions about your work? Do you have resistance to understand what you were doing at that moment, in terms of in terms of grants, in terms of...
Absolutely. Of course, right now I'm emerited. I don't do any experiments any longer. But I still try always to make clear to a wide audience, the general population, of what one can do with genetic engineering is really what nature does all the time. Horizontal transfer of genetic information is only one. I call these natural strategies of genetic variation. One strategy in the genome is to make very local, for example, nucleotide substitution that nature does, because nucleotides that already Watson Crick showed 1953, not in the Nature papers, but later on in Cold Spring Harbor Symposium, in the volume, you see they say occasionally some of the nucleotides have isomeric forms. Isomers means, for example, the nitrogen atom is not fixed. Normal is fixed here, and it may jump here for a short time, perhaps 1 second, going back. But if the replication comes by when it's the isomeric form, A cannot pair with T, but it by chance pairs with C. And if it shifts back, you have a mispairing. In many textbooks, you read that replication gives rise to errors. This is not an error. The enzymes do it properly, but it so happens the property of a nucleotide is that it's not always showing exactly the same three-dimensional arrangement. So one has to really see the laws of nature. Sometimes are unexpected. They are described, but no one takes really that into account. Then the other, the second, that's local, is one stepwise. It can occasionally give an improvement if you change locally some genetic information. The second is you can take a segment of the genome, amplify it, or delete it, or you can take a segment jumping from one place to another, or also what can happen, sometimes the segment is just turned around and gives new fusions. So that's the second strategy. And the third, very important, is the horizontal transfer. And that's a fantastic thing. If you acquire a gene which in some other species functions, and at the condition you have the same language to understand that information, it functions. As far as I know, all living beings have the same language. The universality of the genetic code is behind that. So nature is fantastic.
We agree with that.
And maybe, I may say here, of course, it's Charles Darwin and his colleagues 150 years ago who drove the tree of evolution originally, and saying that those on top of the branches have common origin. And we now say occasionally between branches there is a short transfer of not the whole genome, but just a segment of the genome, relatively small segment. But if that serves you, then you have made a big step advance. And therefore, we have not only common origins, but we have common futures in future evolutionary times.
That's a good thought. That's a good thought.
Right, yeah.
There is the example of the mitochondria. The mitochondria is just a bacteria that that somehow get inside of a eukaryotic cell,
Yes.
and then everything changes. It's not like the monkey went down from the tree. It's like a cell could live
now,
with the energy.
You will now, of course, you can say these have slightly different genetic code rules. And for me, that makes sense. If you have multi-copies in one cell of mitochondria, you need not only one, but you need a certain number. And if you live all your life in that cell, it might be that occasionally from the chromosome or from the nucleus, a gene may get out and trying to get into mitochondria. That's not good for mitochondria. So they must have a slightly different genetic code. Nature is very clever.
Well, thank you very much.
You're welcome.
Was again an honor to have these minutes with you.
Yeah.
And we are going now to sit, and we will leave the students to come, and we will have the conversation with them.
Yes.
Thank you.